right, welcome everybody here in Soils 415 and also on Zoom. This is the penultimate soil, water, and climate seminar for the spring semester. Thank you all so much for your attendance this semester. I think we've had a really great turnout, so it's really great that we can support our speakers and all the great work that's happening in that way. Today, I have the pleasure of introducing our speaker, Dr. Christine Sprunger. Dr. Sprunger is an assistant professor of soil science and rhizosphere processes within the School of Environment and Natural Resources at The Ohio State University. Her research focuses on the intersection of agriculture and the environment. She investigates how climate change impacts crop production, nutrient cycling, soil food webs, and rhizosphere dynamics. In addition, much of her work addresses how crop diversity, perenniality, and reduced tillage contribute to important ecosystem services such as soil carbon sequestration and nitrogen use. Dr. Sprunger also conducts interdisciplinary research with soil scientists to understand farmer perceptions of soil health, adaptation, and climate. And in August, Dr. Sprunger and her lab will be heading to Michigan State University and the Kellogg Biological Station, where they will continue researching soil health in the Department of Plant, Soil, and Microbial Sciences. So we're excited for that new chapter for you, Dr. Sprunger, and we're very excited to have you here today. So with that, I will hand it over to you.
Thank you so much for the invitation and the wonderful introduction. I'm so excited to talk to you all about some of the soil health research that we're doing in my lab. You'll notice a lot of the work that I'll be presenting today is really going to focus on the linkages between soil health and ecological function, with shades of kind of linkages to the agronomic performance piece. So I know I don't have to tell you in this department that there's been a heightened interest in soil health, not only from researchers and people working in extension, but also from farmers across the United States. And before we dive into thinking about why soil health is so critical to study, it's important that we're all on the same page. There's certainly a lot of definitions floating around about what soil health is. I would say the most kind of recognized definition of soil health is that soil health is the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. And one of the, I think, most attractive aspects of just the concept of soil health is it really takes a holistic approach to studying soils. Not only is it thinking about the chemical components of soil, and it also is really interested in understanding the physical and biological aspects of soil as well. And our hope is that by understanding these different components of soil health, we can really understand how healthy soils contribute to really key ecosystem services, whether that be biological diversity, food production, water benefits, or carbon storage. And again, what we've noticed through time is that there's just a lot of interest in soil health at both local and regional scales. For example, farmers want more information from current soil testing. So whenever I talk to farmers in Ohio and Michigan, they often say, listen, the nutrient tests that I'm getting from commercial labs, they're not telling me about the biological aspects of soil or the physical aspects of soil. And so farmers are kind of demanding that researchers give them more information, a more holistic view of their soils. There's also enthusiasm for soil health tests because of their ability to detect recent changes in management. We know that certain indicators of carbon, for example, can detect recent changes in management that often take years to detect when you're just focused on total soil organic matter, for example. And at least here in Ohio, there's a lot of pressure to improve soil health and retain nutrients. And a lot of people believe that there are key linkages between improved soil health and water quality issues. And of course, in Ohio, the major focus is improving the water quality within Lake Erie. Despite the fact that we have a lot of advancements in soil health, you know, the researchers, I would say, in the past 10 years have made leaps and bounds in terms of indicators that are really effective at identifying ways in which soil health is changing in different landscapes. There's still a lot of areas where the field of soil health needs to improve and expand. And I would say one of those is really understanding how soil health responds to management, not only on university research stations, but also on farm. So a lot of the work that I'll present today is actually from on-farm research that we're doing across the Midwest. There's also a need to rigorously vet new soil health indicators, and I'll kind of introduce a couple new indicators of soil health and thinking about how we can kind of integrate them with the other soil health indicators within the soil health framework. Another kind of major focus of my lab is really wanting to understand how we can integrate soil ecology into the soil health framework. And we're actually doing that through the field of nematology and working to understand how nematodes can be used as indicators of soil health. And then lastly, I think there's still a major need for understanding soil health assessments for climate mitigation and adaptation. And I'll talk a little bit about some of the work we're doing at the end in that space. So one of the kind of key principles of soil health is that there are soil health promoting practices that farmers can implement and that we hope will lead to increases in soil health. And these key principles are that farmers need to work on increasing organic matter inputs, whether that be from cover crops or maybe growing perennials. It's also important to diversify soil biota with plant diversity, so extending your crop rotation. It's also important to have year-round ground cover, so you're reducing erosion. And then lastly, it's really important to have living roots throughout the year. And so one of the questions that my lab thinks a lot about is, do these soil health promoting practices actually lead to increases in soil health? And what are the best ways to try to research this empirically? And so one of the questions that we asked using the KBS GLBRC, so the Kellogg Biological Station, Great Lakes Bioenergy Research Center has a long-term trial that was established in 2019. And it's this really nice gradient of low diversity to high diversity. So it has everything from continuous corn, soybeans grown with cover crops. Then you have monoculture perennials like switchgrass and miscanthus that are used as like candidate biofuel crops. And then we have poplar systems, also kind of a dominant monoculture system, but it does have a vigorous understory. So we often think of this more as a polyculture. And then native grasses, early successional and restored prairie systems. And these host a wide variety of different plant species. And so we use this gradient to really understand how soil biological health indicators were changing across the gradient. Now, whenever we talk about soil health, we have to be very clear on the type of indicators that we're using to measure soil health and quantify soil health. And so kind of the three main indicators that I focus on in my lab are microbial respiration. And respiration is really important because it reflects carbon that's most available to microbes, but then it also has the capacity to reflect the amount of carbon that's stored in a given system. So it can be an early indicator of soil carbon accumulation. Active carbon, also known as permanganate oxidizable carbon, is a slightly more processed pool of carbon. So maybe rather than turning over within a day, like respiration often does, epoxy might take a few days, a couple months, but we know that it's slightly more stable pool of carbon than microbial respiration. And then soil protein might be a new indicator for a lot of view, but it is an indicator that reflects the labile pool of nitrogen. And we know it's very strongly associated with soil organic matter cycling. And so in terms of what we saw across this gradient, in this graph A, we're focusing on that mineralizable carbon or microbial respiration. And so the gray bars are annual systems, and then in orange are the perennial monoculture systems, and then in gold are the polyculture perennial systems. And it's very clear that we see this nice increase in mineralizable carbon across the gradient, with polyculture perennials clearly having greater mineralizable carbon relative to the annuals and monocultures. When we look at POXI, or that active carbon value in graph B, we still see differences across the gradient, but the magnitude difference is drastically reduced. And this is, I think, probably the most clear evidence that mineralizable carbon and POX are actually reflecting different pools of carbon, where, 9 years after establishment, they're not, POX is not detecting as large of a difference between these different systems relative to mineralizable carbon. But still, again, we see, especially comparing that polyculture perennial to like continuous corn, there's still significant differences. And then for soil protein, we see trends that closely mirror what we saw with mineralizable carbon. It's picking up large differences between the polyculture perennials and the annual dominated systems. And so just some implications for this first section. Systems with greater perenniality and crop diversity increase soil biological health. Perennial polycultures, aside from restored prairie, increased active carbon relative to annuals, which has important implications for carbon stabilization. Rotated annuals with the cover crop also increased active carbon relative to continuous corn. And this really is a clear indication that soil biological health indicators have the potential to serve as indicators of both nutrient availability and soil carbon stability. But this was, on a long-term university research station. And the next question that we really need to ask is, like, do we see these same trends on actual farmer fields? And I think one of the things, again, that the soil health community is really working towards is understanding soil health on actual farmer fields. So what drives soil health on farmer fields? To address this question, we actually conducted an interdisciplinary research project where we teamed up with social scientists, and we sent surveys to every organically certified corn grower in Ohio, Michigan, Indiana, and Pennsylvania. And farmers were actually offered a free soil health test for completing that survey. Once farmers agreed to accepting a free soil health test, we sent them instructions on how to sample their field. So we asked farmers to select a soil sampling area that was less than 5 acres. And we asked them to take at least 10 cores or slices to a depth of 20 centimeters. And then we had them send composited soil samples with an additional management survey back to us. And then once we received those soils were sieved to 8 millimeters and then ground to 2 millimeters. And then we analyzed the soils for chemical, physical, and biological properties. And then once we were done, farmers received a comprehensive soil test report if they had sent in that management survey. So how did we kind of ask questions about management on that survey? We were really interested in kind of their crop rotation history. So this is just an example of a farmer filling in that they had hay for 3 years and corn. And then we also asked questions about tillage and tillage intensity. And we just had them kind of tick the number of passes that they had and the types of tillage that they were using. So in terms of county representation, again, we had 4 states, 73 counties were represented. In total, we had 195 soil samples as part of the study. In terms of the summary of management practices represented in the study, again, I should note that these were organic farms that were mainly focusing on growing corn. And so no surprise, 87% of farmers were adding manure. Cover crops, there's almost 50-50 with 47% incorporating some type of cover crop. This was really surprising to us. 76% of farmers were incorporating some type of perennial in their rotation. And again, a lot of them were switching between corn and some type of hay. And so that's why that number is so high. And then because again, we're dealing with organic farms, a lot of them, most of them were had some type of tillage operation. So the first kind of question we wanted to ask with this large data set was, what is a good soil health test value? And the reason why we wanted to ask this is because oftentimes when we're talking to farmers about these active carbon values and mineralizable carbon values, they're like, I don't know what this means in terms of a good value. I know what a good soil organic matter value is, but all these other indicators are kind of useless to me unless I know how they compare to other farmers. So the first thing we wanted to do is really have this large database where we could demonstrate distributions of these key soil health indicators. And again, there's a lot of, going on here, but let's just focus on POX, this first graph, and we see really important distributions by texture, where a medium and fine textured soils have an average value of 600 for POX, where a coarse textured soil has much lower. And this is really important because in the soil health test that we gave back to farmers, we were able to say you have an average soil health test relative to other farms with a similar soil type, or you have an above average POX value relative to other farms with similar soil types. And this is, I think, something that is really needed when communicating these new soil health indicators to farmers. So that was something we were just like really excited about from more of a kind of extension and communication standpoint. But of course, the main driver for the study was to understand kind of management and how management impacted soil health indicators. But first, we wanted to kind of understand, really just working with the management survey data, what's the relationship between different types of management practices on these organic farms? Right off the bat, we realized that there are some interesting relationships between crop diversity, tillage, and perenniality. So when we see increases in tillage frequency, we actually see increases in crop diversity. And this makes sense, right? If farmers who are trying to incorporate more crops into their rotation, maybe they're incorporating a cover crop, because these are organically managed farms, they're having to till more to get those different crops into their rotation. But then when we see the length of time that perennials are in the rotation, tillage frequency drastically decreases, which totally makes sense. But keep these relationships in mind as we look at some of these results. So how do different management practices influence soil health in organic corn production? Just focusing on our kind of key 3 soil health indicators, we have POX or active carbon, mineralizable carbon or respiration, and then soil protein. What was kind of surprising to us is while these were weak relationships, they were all slightly negative. And this goes counter to what we generally think about in terms of crop diversity. We generally would hypothesize that soil health will increase with crop diversity and crop, extended crop rotations. But I think what might be happening here is because, again, these are organic farms where more intensive tillage is happening. With an increase in crop diversity, we actually see slight declines in these soil health values. And then when we look at tillage frequency and the relationship between these soil health indicators, we do see, especially with mineralizable carbon, that steep decline in soil carbon with increased tillage frequency. But where we actually see increases in soil health was when we had greater length of perennials present in the crop rotation. So, when the perennials were present for 4 years, we see a significant increase in mineralizable carbon relative to when there weren't any perennials or when perennials were just present for 1 or 2 years. So again, just some really key relationships between perenniality and these soil health indicators. Also interesting to note, of course, that when you have greater perennials in your rotation, of course, you're going to have decreases in overall tillage. So just some kind of concluding remarks on this study. This was one of the first studies to explore management impacts on soil health indicators using a large mail-in survey approach. We're really excited to be able to kind of ask this question on a regional scale. And we found that crop rotational diversity has a complex relationship with soil health indicators, most likely due to increased tillage frequency on these organically managed farms. Generally, when we saw an increase in crop rotational diversity, we saw an increase in tillage and a slight decrease in soil health. When we saw an increase in perennials in the rotation, we see a decrease in tillage and an increase in soil health. I think this study really highlights the importance of conducting on-farm research. We were definitely shocked at the number of tillage passes that occurred within a 4-year period on these organically managed farms. And that's something that probably should be replicated on university experimental trials to understand this interaction a little bit more. And lastly, more large survey assessments are really needed to kind of understand how soil health is impacted by different managements. It could be an effective way to build decision support tools for farmers in the future. So next, I want to talk a little bit about integrating nematology into the soil health framework. So when we look at kind of the soil health indicators that I've mentioned so far, one thing is really absent, and it's that we aren't directly measuring organisms. And there needs to be a better way to understand how soil health addresses biodiversity, and we can't do that without directly measuring different soil fauna. And soil organisms are actually strangely absent from the soil health framework. You look at the different kind of soil health testing options across the United States, and most of them don't directly measure soil organisms. We definitely have indicators that reflect biological activity, such as microbial respiration, but that's not directly measuring who's actually functioning within the soil. And one of the things that has really motivated me to focus on this is that farmers are directly asking for soil health measures that measure these organisms. And so that's one of the things that has really motivated me in my lab to start thinking about which type of organisms might be most useful in understanding soil health. by no means are we the only lab group that are asking these types of questions. Certainly a lot of people are thinking about bacteria and fungi and how they might be able to be incorporated within the soil health framework. And then we have colleagues at Michigan State University that are also thinking about how nematode communities can be incorporated within soil health management outcomes. But one of the reasons why we think nematodes are a critical organism to measure in terms of soil health is just where they're placed within the soil food web. So just as a refresher, nematodes are specialists that are extremely abundant within the soil community. And one of the really interesting aspects of nematodes, because they're specialists, it can actually tell us a lot about what's happening downstream within the soil food web. So there are nematodes that feed on only fungi. There are nematodes that only feed on bacteria. There are nematodes that actually feed on each other. Those are the predators. And then most people are really familiar with the plant parasitic nematodes that feed on plant roots. And so oftentimes when we talk about nematodes, we definitely think, oh, that's a bad thing. They are parasitic. Well, actually, most nematodes are very beneficial. And those are the ones that are feeding on bacteria, fungi, and each other. And when we quantify nematodes, not only can we learn about the structure of the soil food web, we also know that they are really linked to ecological function. So once you identify nematodes, you can calculate different indices that reflect ecosystem function. So for example, the structure index reflects trophic complexity and diversity. And really you can think of this as kind of like an overall like biodiversity metric. And then you have the enrichment index, which really pinpoints nematodes that are key in terms of nutrient cycling. So oftentimes the enrichment index will increase when you have a system that's really high in organic matter, really it has a really high nitrogen value. And then you have the channel index, where you have fungal and bacterial decomposition pathways. So that's really important in terms of thinking about just decomposition in general and how that's linked to nutrient cycling as well. And then you have the basal index, and based on the ratio, this can be an indicator of the amount of disturbance or the overall health of an ecosystem. So how might we use these nematode indices in practice? So in this left quadrant, we're comparing the enrichment index to the structure index. And this was developed by Howard Ferris, was a professor at UC Davis. And when you have a high enrichment index, but a low structure index, you have a system that might be extremely disturbed. When you have a low enrichment index and a low structure index, you could have a system that's very degraded. But then when you move to the right, when you have a high enrichment index and a high structure index, you have a system that's maturing, nitrogen enriched. And then when you have a system that is low in enrichment but high in structure, it's a matured fertile system. And so just kind of overlaying some points from a research study that I conducted at Michigan State University, where we were comparing perennial kernza grown organically and then annual wheat grown either organically or inorganically. We can actually see that this kind of approach works quite well, where you have perennial kernza and annuals grown with an organic source. They're in the more maturing or matured quadrat, but when you have an annual system that's just receiving inorganic fertilizer, it's going to be a more degraded system. So again, this just demonstrates that nematodes can be really effective in not only demonstrating just soil food web structure, but making linkages to ecosystem function as well, which is why we think that this could be a useful tool in terms of thinking about soil health. So the next question that I'll pose is, how can we integrate nematode community data with other soil biological health indicators to understand soil health outcomes? Now, to do this, we actually used kind of a very kind of advanced quantitative approach. And we used an exploratory factor analysis. This is a quantitative solution born out of multivariate statistics to identify the underlying relationships between measured variables. And this figure here is actually an example from a paper that's actually led by Jordan Wade, just in press, and there's a lot of people from the soil health community that are on this paper. But one of the things that we're asking is, how can we determine which soil health indicators are most relevant to test within a given system? And not only are we interested in understanding which indicators might be most useful, we're also interested in understanding the key relationships between them. And so by using this approach, my grad student came to me and said, let's do this with nematodes as well. And I was like, well, tell me why we need to do an exploratory factor analysis. Can't we just do correlations or something simpler? And she said, think of all the soil health indicators that we're working with. Correlations are only telling us the relationship between two factors, and it's way more effective and meaningful to understand how these indicators are all related and how they might be influencing soil health outcomes. So that's exactly what we did. And we think it's a key approach for identifying soil health outcomes and could be used for sustainability goals in the future. So I'll kind of just walk through exactly what we did, because again, this is like a new kind of approach. I would say new to soil health. It's very common kind of in fields of psychology and social sciences. So we'll walk through it together. So basically what we did was we took all of the data that we have on nematodes. So anything that we've collected from on-farm surveys and all of the work that we've done using long-term trials here in Ohio. And using this model, what we have is all of these different soil health indicators. So the nematode indices, nematode feeding groups, enzyme activity, pox, protein, and respiration, which I've talked a lot about already, and then organic matter. And we fed this all into the exploratory factor analysis. And through this quantitative analysis, what we get are these different factors. And these factors are basically what we're calling soil health traits or soil health outcomes. And what we're trying to understand is how these different soil health indicators are loading on to these different factors. And then a really kind of useful aspect of this approach is that we're able to name these factors. So factor one, factor two, that doesn't really mean anything from a soil health perspective, but we can actually name them based on the different indicators that are loading onto those factors. And then you can do your typical kind of ANOVA analysis with these factors. But really, I want to focus just on this objective one. And again, we'll take it slow and walk through it together so people don't get lost. So with this factor one, how we read these are the circles are the factors, and then the black arrows represent something that loaded positively and significantly. And the red arrows are something that loaded significantly, but the relationship is actually negative. So again, with this factor one, positively loading was aryl phosphatase, an enzyme. fungivore feeding nematodes, and then poxy or active carbon. And then the negative significant loadings were plant parasitic nematodes, mineralizable carbon, and protein. And what we decided to actually name this factor was a fungal organic matter processing pathway. We oftentimes associate kind of fungal feeders and fungi with RO phosphatase or phosphorus cycling. And interestingly enough, we also oftentimes associate fungal kind of pathways with slightly more stabilized pools of carbon, which would be POX. And it makes total sense that mineralizable carbon is kind of negatively loading and loading opposite to what POX might in this scenario. And then for factor 2, we decided to name this rate of nutrient cycling. Again, we had several different type of enzymes, and then we also had omnivores and predators, which again are often associated with higher rates of nutrient cycling. And then we had this third factor where we just had enrichment or nematode indices loading. So we have the maturity index, the structure index, and then the enrichment index. And given that the maturity index and structure index were both significant and positively loaded, we decided to call this factor trophic complexity. And then lastly, we have, again, a fourth factor with just nematode indices loading. And we have positively loaded enrichment index and structure index, and then negatively loaded is the basal index. And again, that represents disturbance. And one thing that I really want to highlight here, or I guess two things. One, organic matter is not to be seen on this. Soil organic matter, the most commonly used indicator that's available to farmers, didn't load at all. It means it wasn't associated highly enough with any of these indicators to load onto the factor. So that's kind of a major issue. And then two, it's interesting that the nematode indices were kind of separate from the nematode feeding groups. The nematode feeding groups seem to kind of integrate well with these other soil health indicators, things like POX, things like mineralizable carbon. So some key findings from this and how we're kind of thinking about how to move some of this work forward is that nematode feeding groups share underlying soil health traits with key soil biological health indicators. This means that nematodes should be integrated with other soil health indicators and used in soil health assessments. Soil organic matter that was determined via loss on ignition did not integrate with other soil biological health indicators, which was a major surprise to us. And the feeding groups tended to be more closely associated with soil biological health indicators compared to the nematode indices. So another way to interpret this is that nematode structure seemed to be more integrated with other soil biological health indicators relative to these nematode indices, which their sole purpose is to try and reflect ecological function, which may mean that nematode indices actually aren't as useful in terms of reflecting function as we once thought. And then lastly, developing regionally relevant nematode indices, I think, could be key to linking both soil biodiversity and ecological function to soil health outcomes. And then lastly, I just want to talk a little bit about some of the current research that we're doing. As you heard, my lab's moving to Michigan State University, where we're already doing a lot of research. But one of the major research projects that we have is a rainout shelter experiment to explore drought impacts on soil food webs, especially nematodes, using the KBS LTER. And then we also have a large study that focuses on the impacts of flooding in corn-based systems, and we have several trials across Ohio. My colleague Alex Lindsay is focusing on the agronomic aspect of this and really wanting to understand how flooding is impacting not only corn production, but also the different ways in which we might need to apply nitrogen fertilizer in maybe needing to adapt the way that we apply fertilizers based on flooding events. And then my lab is focusing more on the rhizosphere dynamics and soil health aspects. So again, we're looking at nematodes and other soil biological health indicators and really understanding, can these different indicators be used as assessments within systems that are experiencing climate extremes. So just some overall conclusions, systems with greater diversity and perenniality, enhanced soil biological health in long-term trials in Michigan. On-farm soil health assessments will be critical moving forward as we work to incorporate soil health data into management recommendations. And then lastly, nematode feeding groups seem to integrate well with other soil biological health indicators and should be included in routine soil health assessments. And with that, I just want to acknowledge the members of my lab. They're the ones doing all of the major field work and lab work. And then I want to thank my funding sources and key labs that we collaborate with. I'm happy to take any questions at this time.
Great, thank you so much, Christine. And as usual, if folks are on Zoom and want to either type their question in the chat, Amy will be monitoring that, or you can unmute and ask a question, and then those in the room want to kind of get closer to the mic, that would be great.
I got a question.
Okay.
Thanks for the, can you hear me?
Yes, I can.
Oh, great. Very interesting talk, and thanks for coming to the to our department to present. I was really interested in your nematode data. It's very interesting. I'm wondering how you actually measured the feeding groups. Did you differentiate between the parasitic nematodes versus the non-parasitic? And how did you do that? I mean,
yeah, no, that's a really great question. So the way that we identify nematodes is through microscopy. So it's very laborious. So we first count nematodes. So we just take 100 grams of soil and then extract them using Bierman funnels, and then we count them. So sometimes we have upwards of 1,000 nematodes within a single sample. And then my grad student counts them and then identifies them. And so I actually identify them to the family level. And then based on the different families, we're actually able to calculate, the number of feeding groups that are present, and then we can also calculate those nematode indices. And Howard Ferris actually came up with this really awesome open source software called Ninja. And so once you have counts and the identification down to at least family, sometimes people go to genus, then you can start to categorize the feeding groups that are present, but also calculate those indices as well.
Yeah, interesting. So the plant parasitic nematodes, is that a particular feeding group then? Is that
exactly? Yep. And so the kind of 5 feeding groups are the plant parasitic, the fungivores, bacteriivores, the predators, and then we also have omnivores. Those are not specialists. They'll kind of just feed on whatever's present and available to them.
And were the plant parasitic nematodes correlated with in particular correlated with any soil health indicator?
Yeah, so if we look back at this value here, the plant parasitic feeding group was negatively loading onto this fungal organic matter processing pathway. So it was actually more associated with mineralizable carbon and protein, which are kind of mineralization processes within the soil, which is actually interesting. That makes sense. If they're kind of disturbing the roots of a given system, maybe those roots are maybe turning over a little bit more quickly and maybe kind of providing a burst of nutrients, rather than the fungivores in that case were more associated with active carbon.
Thank you.
Great question.
We have a question in the chat from Gabriella. Are nematodes assessed through DNA sequencing, and how do you classify them into functional groups? So maybe you covered some of that in the previous question.
Yeah, so that's an excellent question, because you know, it's a laborious process to identify nematodes through microscopy. The natural question is, why can't we use molecular methods? Well, people are developing primers, and what we've seen is that there's only about 30% accuracy in terms of identifying nematodes via molecular approach. And so we're not quite there in terms of solely relying on molecular methods. That's definitely something that nematologists, especially at Wageningen University in the Netherlands, they're working on that. But the most accurate way to quantify and identify nematodes right now is through microscopy.
Anna, do you want to?
Yeah. Hi, Christine. It's Anna.
Hi, Anna.
I have a question. So I was early on struck by the tight correlation between your soil health indicators and organic matter. Noting, I think it was in the one where you separated things by texture, that everything was kind of following the same pattern as the organic matter, like the mineralizable carbon and nitrogen and such. But then later it looks like, oh, you know, organic matter doesn't fall out in this more sophisticated multivariate approach. So where do you think the role is for organic matter? Again, this really common, cheap, easy, you know, recognizable indicator is within all these other possibilities.
Yeah, I think that's such an important question, especially given that you're right. Organic matter is literally the indicator that's most accessible to farmers. And I think we've always known, especially even going back to some of my dissertation work, is that organic matter is like hugely important in just kind of reflecting the overall soil health status to like for a given farmer. But when you're trying to detect, say, recent changes in management, or trying to understand how these other more kind of sensitive indicators are related to this very important process, organic matter kind of falls off. And I think it's because of organic matter being determined via loss on ignition. And I'm wondering if, had we used like soil carbon in that model, would we have had the same effect? I just think that the loss on ignition kind of model is just not as sensitive to kind of all the other noise that's happening within these systems. But it's something we certainly need to figure out, because again, it's the indicator that's most available to farmers.
Thank you. So you think soil carbon might have a chance. That's good to know. And then I'm curious too, in terms of price and accessibility and, you know, actually using nematodes for farmers. Do you think that you could get the cost of nematode analysis down enough and relate it to management decisions enough to make it part of a standard soil test? Like, what are the steps to getting there?
Yeah, and so that's why the Gabriella's question was so critical. I think we'll need molecular approaches to help with at least the kind of rapid assessments of nematodes. I will say we can look to European countries, especially the Netherlands, where they're actually already incorporating nematodes into soil health tests. And they actually have labs where people are they can rapidly identify nematodes. That's how quick they are at being able to assess nematode communities. And they're actually giving that information to farmers, and farmers are already basing management decisions off of those tests. So I think it's one, if we're going to stick with microscopy, it's building that skill set here in the United States. And two, also like continuing to research how molecular methods can be used.
Great, thank you.
Fabian, do you want to go next?
Sure, thank you. Yes, a great presentation. And this is maybe a little bit of a follow-up to what Anna was asking, because I found that really interesting that the organic matter didn't seem to be that important in terms of helping us. But we all know that, I mean, for millennia, people have looked at soils and whether soils are productive or not, mostly based on color, the amount of organic matter. And I was wondering if it's related to the fact that maybe the biggest amount of organic matter, the biggest pool, is kind of recalcitrant, and so it's not very indicative, I guess, of what's happening at the moment. But it is important in terms of, you know, all the stuff that we know about organic matter, how it helps everything in the soil.
100%. I think you're spot on that. I think because we're looking at these sensitive indicators that are reflecting labile pools of carbon, of course, they're not necessarily most associated with soil organic matter, when, yeah, they're definitely more associated with soil organisms, which we know are really critical in influencing that labile soil or that labile carbon pool. And so I think that you're right, that organic matter that is reflective of the total amount of organic matter in the soil, and it's just not responsive with these other indicators that are picking up kind of more rapid pools in the soil.
Katsutoshi?
Yeah, thank you so much for the presentations, and Christina, and thanks so much for interest in the research projects as well. I've done a lot of soil health quantification projects, and I really wanted to learn more about the your research in details. So you use the if I remember correctly, exploratory factor analysis. Is that what you use for calculating the soil indicators, soil health indicators, if that's the case? So I'm not a statistician, but the is a kind of simplified version of a PCA, and picks up the variables that correlates each other as a strong variables to be considered as a model, for the factor. So my question is, what if there's a important, conceptually important soil variable, or microbes that are really significantly affect the soil functions, nutrient cycle, for example, but that does not correlate with the other variables. Probably this approach might not be picking up the variable. So my question is, based on what you have accomplished, so many different techniques, you incorporate a lot of variables together for the soil health assessment. I'd like to know what will be the next step, in terms of this factor analysis. So yeah.
No, it's such a great question, and you're absolutely right. I mean, we're not going to disregard soil organic matter because it didn't load onto these different factors. And I think the next thing that we need to do is go back and do another empirical study, maybe in a different spot, like we're doing at the Kellogg Biological Station in Michigan, and understand if these different relationships continue to hold. Do we start to see the same type of relationships and the same type of indicators integrating with one another in the same way? One thing that we're actually really interested in is all these different enzyme activities and how they're associated with these different feeding groups. That's not something that we know a lot about in the literature. And so I think, that's something that we can take from the study and then develop other experiments to fully understand what those relationships look like.
It sounds really great. I can't wait to have like I don't know, to see like an incubation test or
Yeah, exactly.
yeah, that's very interesting. Thank you so much.
Thanks for the question.
Questions for Christine? See any more hands raised? If not, Christine, thank you so much again for your time and your super interesting presentation, sharing your work, and we wish you luck on your move to your new position.
Thank you so much. I really appreciate everyone's attention, and thanks so much for the invitation. It's been fun to chat about soil health with you all.
All right, thanks again, all. That concludes Spring Soil Water Climate Seminar Series. Thanks, everyone.